Welcome student for today's lecture. Student, in last lecture we have seen that is about the translation process we have start. And in the translation process we have seen the requirement which is required for the translation process. So student, today we are going to see that is about the translation process. Actually the translation, how the translation takes place. So we know that is the translation, it is the process that is here the synthesis of the polypeptide chain takes place by the mRNA.
Translation is the process whereby the base sequence of a messenger RNA is converted into the amino acid sequence of a polypeptide. This process involves an mRNA, tRNAs, amino acids, a ribosome, and other accessory proteins. mRNA consists of three main parts. The portion from the 5' end to the translation start codon is the 5' untranslated leader region, more simply called the leader. The central portion is called the reading frame. It commences with the start codon, AUG, followed by a series of 3 nucleotide codons, each of which specifies an amino acid, and it ends with one of the stop codons, UAA, UAG, or UGA. The final portion of the mRNA consists of the section from the stop codon to the 3' end, called the 3' untranslated trailer sequence, more simply called the trailer. mRNAs vary in length because of differences in all 3 parts of this molecule. Transfer RNA functions to bring amino acids to the growing polypeptide during translation. It consists of a single RNA chain of about 75 to 90 nucleotides, each tRNA having a different sequence of bases. After it is transcribed, the tRNA folds up into a characteristic cloverleaf structure. The anticodon contains a 3-nucleotide sequence that is complementary to a codon on the mRNA. The amino acid is attached to the 3' free end at the top of the cloverleaf. The ribosome assembles amino acids into a polypeptide. Ribosomes consist of 2 parts, a large subunit and a small subunit. Ribosomes are made of ribosomal RNA and various proteins. The active ribosome contains 3 tRNA binding sites, namely E, exit, P, peptidyl, and A, aminoacyl. In prokaryotes, translation initiation occurs in 3 steps, as follows. First, the small ribosomal subunit, 30S subunit, complexes with 3 proteins named initiation factor 1, initiation factor 2, and initiation factor 3. A molecule of GTP also attaches. The ribosome IF-GTP complex recognizes a sequence in the leader, upstream of the AUG start codon. This sequence is called the ribosome binding site, or the Shine-Dalgarno sequence, after its discoverers. This sequence is complementary to a sequence at the 3' end of the 16S ribosomal RNA of the 30S ribosomal subunit, and serves to align the ribosome on the mRNA in its proper reading frame. Second, translation begins at the start codon AUG, a few bases downstream of the ribosome binding site. The first AUG in proteins specifies a modified form of methionine. This modification consists of the addition of a formyl group to methionine, making it formylmethionine. The anticodon of this initiator tRNA containing FMET complements AUG. Upon binding of FMET tRNA to mRNA, IF3 is released, with the remaining assembled components called the 30S initiation complex. Lastly, the large ribosomal subunit, 50S subunit, binds to the 30S initiation complex. GTP is hydrolyzed and IF1 and IF2 are released. The final complex is called the 70S initiation complex. The FMET tRNA is now situated in the P site of the ribosome, with its anticodon hydrogen bonded to the mRNA start codon. Initiation is now complete and elongation
Okay, so here the translation, it mainly completes in the three steps. That is, the translation, it mainly completes through the three steps. So first step is the initiation. Initiation of the polypeptide chain. Second step is the elongation. Elongation of polypeptide chain. And third is the termination. Termination of polypeptide chain. Termination of polypeptide chain. And so this polypeptide chain म्हणजेच काय त्याला म्हणणार आपण प्रोटीन. दॅट इज द proteins, which are the proteins. So these are the three steps, and through these steps here the translation process completes, and here the translation, that is the synthesis of the protein, that is the polypeptide chain of the amino acid, takes place. Okay, so student, today we see that is the first step, that is the initiation. So see, that is here the initiation means here there is the translation process. The process of the translation here starts. So what happens during the initiation? So the first step of the initiation is the activation of nucleotide. So here the first step and the most important, that is the activation of nucleotide. Activation of amino acids. That is here the amino acids, it gets activated. So activation of amino acids. So here in the cell cytoplasm, we know there are the 20 amino acids, and these 20 amino acids, they get activated. So how they get activated? So see, that is suppose here there are the amino acids. Okay, so there are the different 20 types of the amino acid. For example, it is the AA1 amino acid, it is the AA2 amino acid, here will be the third amino acid, here will be the fourth amino acid, like this. So there are the different amino acids. Okay, so these are the amino acids. These are the amino acids. So these amino acids, they get activated. So how they get activated? That is, they get activated by the ATP. So here the ATP, that is ATP energy, is required for the activation of amino acids. Okay, and here these are activated in presence of enzyme amino acyl tRNA synthetase. The enzyme is amino acyl tRNA synthetase. Amino acyl tRNA synthetase. It is the enzyme. So in presence of this enzyme and with the ATP, here the amino acids gets activated, and there is the formation of amino acyl tRNA complex. So see, that is here how the amino acid tRNA complex is produced. So see, that is here the tRNA structure, that is the hairpin model of the tRNA. If we see, that is, it is having the two ends, that is here the two free end we have seen, that is this end, it is the 5' end at which here the 3 codons, that is the free nucleotides are there, that is here the CCA end, it is the CCA end. So at which end here there is the free hydroxyl group is present, and this due to presence of this hydroxyl group, that is activated hydroxyl group, here the amino acid gets attached at the 3' end. So here the one amino acid gets attached at the 3' end. Here is the 5' end at which here the guanine is present. Okay, and there are the anticodons on the anticodon loop. So here this tRNA with the amino acids, that is nothing but the amino acid tRNA complex. It is the amino acid tRNA complex. Okay, it is the amino acid tRNA complex, or it is also known as charged tRNA. यालाच काय म्हटलं जातं, charged it is also known as the charged tRNA molecule. Charged tRNA molecule. It is also called as charged tRNA molecule. So see, that is here amino acid, it gets activated, that is amino acyl tRNA synthetase enzyme. हे काय आहे, एन्झाईम आहे, synthetase enzyme. And here there is the formation of the tRNA, that is amino acid tRNA complex. काय तयार होईल, amino acid tRNA complex, आणि त्यालाच आपण काय म्हणणार, charged tRNA. त्यालाच काय म्हणणार आपण, charged tRNA. So here activated amino acid, it mainly gets attached at the 3' end of the tRNA, that is here CCA end. Okay, it is the CCA end. हे लक्षात ठेवा, at which end the amino acid gets attached, that is the CCA end. Okay student, then next see, that is about the how the amino acids they are that is in the ribosomal unit enters, and how the protein synthesis takes place. So see, that is here the protein synthesis takes place, that is in the ribosomes. So here there is the initiation process. So what is about the initiation? So initiation, how the initiation takes place. So during the initiation, see here there is the mRNA. Okay, it is the mRNA. On this mRNA, here is the 5' end, and here is the 3' end. We know that is at the 5' end there are the initiation codons. So which are the initiation codons? That is AUG. So see here there is the initiation codon, that is the AUG. So here before this initiation codon there is the UTR region. So from this region to this region there is the UTR region. There is the UTR region. So UTR region is again here, that is after the termination codon. So see here, that is here is the from this region to the this region, here is also the UTR region. UTR region. So this UTR region, it is the before initiation codon and after termination codon. After termination codon. So for example, see here, that is the termination codon UAA. Okay, UAG and UGA. So here here is the UAA termination codon. So after this termination codon, here is the UTR region. So see here, that is at the 5' end where the UTR region is. UTR region is before initiation codon. हे इनिशिएशन कोडॉन AUG हे इनिशिएशन कोडॉन आहेत, त्याच्या पूर्वी before initiation codon UTR region is there. So what is mean by the UTR region? UTR region means here untranslated region of mRNA. UTR region means what? Untranslated region of mRNA. UTR region means what? Untranslated region of the mRNA. So here this untranslated region of the mRNA, that is the UTR region, so it mainly present, that is the before initiation codon and after the टर्मिनेशन कोडॉन. हे टर्मिनेशन कोडॉन आहे 3' एंडचे, त्याच्यानंतर हा रिजन येईल, दॅट इज द अनट्रान्सलेटेड रिजन ऑफ mRNA. ओके, UTR रिजन. आणि AUGच्या अगोदर म्हणजे इनिशिएशन कोडॉनच्या अगोदर आपल्याला हा UTR रिजन येईल. ओके, सो सी दॅट इज हिअर इन द प्रेझेन्स ऑफ मॅग्नेशियम आयन. कोणत्या आयनच्या प्रेझेन्समध्ये? In the presence of the magnesium ion, what happens, that is the smaller subunit of the ribosome is gets attached to the UTR region. आता हा UTR region आहे, ठीक आहे. इथे इनिशिएशन कोडॉन, त्यानंतर अजून एखादा इथे कोडॉन असेल, ठीक आहे, त्यानंतर अजून कोडॉन असतील आणि त्यानंतर मग हा लास्ट टर्मिनेशन कोडॉन येईल, ठीक आहे. टर्मिनेशन कोडॉन नंतर पुन्हा UTR region येईल. मग mRNA की ज्याच्यामध्ये UTR रिजन आहे, ॲक्च्युली या UTR रिजनवरतीच काय होतं, तर रायबोसोमचा स्मॉलर सब युनिट काय होतो, बाइंड होतो. आता स्मॉलर सब युनिट आणि लार्जर सब युनिट हे आपण लास्ट लेक्चरमध्ये स्टडी केलेलं आहे. तर स्मॉलर सब युनिट हे इथं सुरुवातीला बाइंड होईल, दॅट इज ॲट द UTR एंड. सो सी हिअर देर इज द स्मॉलर सब युनिट, सो इट गेट बाइंड्स like this. It get binds, that is the at the UTR region. So students, see that is here the UTR region, that is at which here the smaller subunit of the ribosome gets attached. So it is the smaller subunit of the ribosome. आणि हा UTR region ज्या mRNA मध्ये नसेल, this mRNA is unable to transcribe. म्हणजे लक्षात ठेवा की the mRNA which is not having UTR region, की mRNA की ज्याच्यामध्ये UTR region नाही, this mRNA is unable to transcribe. तो mRNA ट्रान्स्क्राइब होऊ शकत नाही, म्हणजे त्या mRNA पासून प्रोटीन सिंथेसिस होणार नाही. लक्षात येते? सो हिअर द UTR region is most important, that is at which here the smaller subunit of the ribosome it gets bind. Then here the smaller subunit when gets attached, it acquire the first कोडॉन्स. म्हणजे आता जनरली फर्स्ट 2 कोडॉन्स हे ॲक्वायर करेल. म्हणजे बघा, जेव्हा हा स्मॉलर सब युनिट अटॅच होईल, इनिशिएशन कोडॉन हा पहिला कोडॉन आणि दुसरा कोडॉन कोणता त्याच्या सलग असेल तो दुसरा कोडॉन असेल, तिसरा चौथा असा. मग पहिले 2 कोडॉन हे काय करेल, हा स्मॉलर सब युनिट काय करेल, ॲक्वायर करेल. ओके, सो हिअर द स्मॉलर सब युनिट दॅट इज इट मेनली ॲक्वायर्स अटॅच अँड ॲक्वायर द 2 फर्स्ट कोडॉन्स, पहिले 2 कोडॉन्स, हा कोडॉन आणि हा कोडॉन. सो द फर्स्ट 2 कोडॉन्स they are acquired by the smaller subunit. Then here the larger subunit having the 3 sides, it gets attached like this here. So see that is, it is the larger subunit. Larger subunit of the ribosome. Here is the smaller subunit of the ribosome. So in last lecture we have seen that is the larger subunit, it mainly made up of the 3 sites. Which are the 3 sites? That is, here is the first site, that is the A site, through which here the amino acid tRNA complex it is accept. Okay, then another site, that is the second site is the P site. It is the P site. And there is the third site is the E site, that is the exit site. E site is the exit site. So there are the 3 sites. So in last lecture we have seen that is through the A site here the tRNA complex it is accept or enter through the A site, then through the P site polypeptide chain is released, and here through the E site that is the empty tRNA, म्हणजे जेव्हा इथे अमायनो ऍसिड दिला जाईल तेव्हा empty tRNA हा या E site मधून काय होतो, it gets released, that is through the E site. Okay, so student, during initiation what happens, that is here the charged tRNA molecule it mainly enters, that is through the P site. So first tRNA, जो charged first tRNA असेल, it mainly enters through the P site, and remaining tRNA they enters through the A site. Okay, they are accept through the A site in the ribosome. But in the eukaryotic cell and also in the prokaryotic cell, the first tRNA, that is which mainly bind the AUG, that is the initiation codon, this tRNA enters only through the P site. कोणत्या site मधून enter होतो तो P site. So see, that is here, suppose here is the activated tRNA. Okay, so it is the activated tRNA. See here is the end, that is the CCA end, at which here the amino acid is attached. Here is the amino acid, and there are the 3 anticodons. Okay, 3 anticodons. So it is the activated, that is the amino acid tRNA complex, charged tRNA म्हणतो त्याला आपण किंवा चार्ज्ड अमायनो ऍसिड. ठीक आहे, तर हा ऍक्टिव्हेटेड tRNA जो आहे हा ऍक्टिव्हेटेड अमायनो ऍसिड कॉम्प्लेक्स हे काय होतं तर इट गेट्स एंटर द फर्स्ट अमायनो ऍसिड, इट एंटर्स इनटू थ्रू द पी साइट, आणि राहिलेले बाकीचे जे आहेत ते कुठून एंटर होतील, या ए साइट मधून एंटर होतील. आता इथं पी साइट मध्ये जेव्हा एंटर होतील तेव्हा काय होतं बघा, तर द फर्स्ट tRNA amino acid complex, first tRNA amino acid complex or the charged tRNA म्हणतो त्याला आपण, charged, it is the charged tRNA molecule. So in the eukaryotic cell, in eukaryotic cell, here the amino acid first carried by the tRNA is the methionine. Is the methionine. म्हणजे कोणता अमायनो ऍसिड इथं कॅरी होईल, मिथिओनाइन. while in prokaryotes, जर आपण प्रोकॅरियोटिक सेल मध्ये पाहिलं, in the prokaryote, that is the first amino acid which is carried by the tRNA, that is formyl methionine. It is the formyl methionine. It is the formyl methionine. So see, that is here in the eukaryotic cell. So first amino acid it is carried, it is the methionine, and this methionine which is carried, that is by the eukaryotic cell. So see, that is here the tRNA, that is activated tRNA, it enters through this P site, okay, and it binds the first initiation codon, that is the AUG. So what will be the anticodons? आता या tRNA वरती इथं काय असणार आहेत? anticodons. So what will be the anticodons? That is, there are the complementary anticodons. So here is the adenine, then here the anticodon of the tRNA will be. आता याला complementary anticodon इथं कोणता असेल? That is the uracil. Here is the uracil, here will be the adenine, and here is the guanine, here will be the cytosine. So here this tRNA which carries the methionine, म्हणजे इथं समजा हा methionine हा amino acid जर हा tRNA कॅरी करत असेल, this tRNA having the anticodons, that is the UAC. The tRNA having anticodons, that is the UAC. And this tRNA having the anticodons UAC, it mainly carry the methionine amino acid, and this tRNA, activated tRNA, it enters through the P site. कोणत्या साइट मधून एंटर होईल? या P साइट मधून एंटर होईल, and it get bind with the AUG, that is the initiation codon. लक्षात येते? म्हणजे इनिशिएशन कोडॉन याला बाइंड करणारा युकॅरियोटिक सेल मधला कोणता tRNA असेल की जो tRNA कोणता अमायनो ऍसिड कॅरी करेल? मिथिओनाइन. मग प्रोकॅरियोटिक सेल मध्ये तोच अमायनो ऍसिड कोणता आहे? फॉर्मिल मिथिओनाइन. कोणता अमायनो ऍसिड? फॉर्मिल मिथिओनाइन. अँड दिस फॉर्मिल मिथिओनाइन कॅरी करणारा tRNA इट एंटर्स थ्रू द P साइट. लक्षात येते? आता त्यानंतर जे tRNA येतील, these tRNA, these are enters through the A site. So remain that is here the only first tRNA which codes the initiation codon. This tRNA only enters through the P site. Okay, and remaining are enters through the A site. Okay, student. So here in the initiation, that is here we have seen that is how the initiation takes place. That is the activated tRNA, it mainly enters through the P site of the ribosome, that is the larger subunit. म्हणजे larger subunit मध्ये आपल्याला माहिती आहे 3 साइट आहेत, A site, P site and E site. आणि इनिशिएशन मध्ये पहिला अमायनो ऍसिड जेव्हा एंटर होईल, इन द यूकॅरियोटिक इट इज द मिथिओनाइन, अँड इन द प्रोकॅरियोटिक इट इज द फॉर्मिल मिथिओनाइन. ओके, सो स्टुडंट हियर इट इज द इनिशिएशन ऑफ द polypeptide chain. Okay, so student, so today we stop here.